Chapter 11, the Nervous System Integration and Control, Screencast 3. Now, this screencast is going to focus on the concept of chemical synapses and synaptic transmission. But first, we need to talk about neuroglial cells. Now, remember, when we talk about neural tissue, there's two general types of cells. Neurons, which there aren't as many of, are those that actually generate the electrical signals. And then there's the support cells, these neuroglial cells, and they actually make up approximately 80% of the nervous system. So we want to think of them as support. They're going to protect. And the most important thing here is they do not transmit or generate action potentials. That is strictly neurons that have that capacity. Now, depending upon what part of the central nervous system they are, they're going to be different. Within the peripheral nervous system, we have Schwann cells. In the central nervous system, we have oligodendrocytes. So these are the primary types of glial cells that we will see, and it's very dependent upon the branch of the nervous system. So once again, Schwann cells are found in the peripheral nervous system. Oligodendrocytes are found in the central nervous system. Now, a couple of things that I want to point out here because I want to make sure that you're comfortable with the general anatomy of a neuron. So these are dendrites, okay? So these are the receiving end of a neuron. There's the cell body, that's where the nucleus is going to be. And then we have the transmitting end, which is the axon. And what we have here is actually a myelinated axon. And that's one of the things that's going to increase conduction velocity. And what we have is known as saltatory conduction. And notice what's happening here. Okay? So the wave of depolarization is going to go from one of these areas, which is a node of Ranvier, to the next. So you're not depolarizing the axon. You're basically leapfrogging from one node of Ranvier to the next. So that allows you to go down the length of the axon much more quickly. So once again, the two things that are really impacting conduction velocity are axon diameter and whether or not the axon is actually myelinated. So remember, myelin is not continuous. And the spaces in between myelin are known as nodes of Ranvier. Okay? So Schwann cells make up the myelin sheath in the peripheral nervous system. And as I said before, you have this saltatory conduction that's going to result in increasing conduction velocity. Now, another role that Schwann cells are going to play is to help cells that are damaged or severed and allow axons to regenerate. That's very limited capacity. Okay, I want to make sure that I really reinforce that. When we have a damaged neuron, okay, there is some wiggle room for regeneration, but it is not significant. And we know that if you know someone that's had a spinal cord injury and they're paralyzed because of that, depending upon the location of the lesion. And oligodendrocytes, they're actually responsible for forming the myelin sheath in the central nervous system. So you want to be comfortable with the characteristics of these, and most importantly, okay, the role of myelin. Now, there are two significant disorders that are associated with degeneration of the myelin sheath. We look at multiple sclerosis. Now, this is a progressive damage to myelin sheaths within the brain and spinal cord. You may know some people that have multiple sclerosis. This is actually known as an autoimmune disease. And autoimmune diseases are those where the body starts to attack its own cells. And so MS is a really good example of that. And common symptoms associated with this are weakness, visual impairment, and incontinence. Now, ALS, and some of you may have participated in the water bucket challenge a few years back, and that was actually to promote, let people know about this particular disease. This is a devastating disease. It's progressive in nature, and it actually is specific to the myelin sheaths in the motor area of spinal cord. So what we see here is a weakening and wasting of skeletal muscle. Stephen Hawking's, that was the disease that he had. I've had a couple of friends that have had ALS, And it's a truly devastating disease because what you're seeing primarily is decline in motor function and nothing in cognitive function. So you're completely aware of what you're experiencing. And to me, this is a very devastating disease. Now, the next thing that we want to look at is how do neurons communicate with each other? And the majority of these is through synaptic transmission. And so what's happening here is a neuron is going to interact with either another neuron, a muscle cell, or a gland. So a synapse is the connection between two. Okay, and when we talk about this process of synaptic transmission, really what we're looking at is the transmission of an impulse from the sending or presynaptic neuron across the synaptic cleft. So this is specific to chemical synapses, and that's what we're going to focus on primarily. And a synaptic cleft is a space between the presynaptic neuron and the postsynaptic neuron. So these are not physically connected together. That's really, really important. And then to the postsynaptic neuron, which is the target, okay? And eventually what we're looking at here, because we're talking about chemical synapses, we're really looking at neurotransmitters. Okay, so this is where we're kind of looking at now. And remember, the ultimate goal or role of that action potential was to release neurotransmitter. Here, this is something that you're going to need to be comfortable with the various steps. So the first step is you have to have an action potential. Okay, so that action potential is going to propagate, okay, down. And then what's going to happen here is those action potentials are going to trigger the opening of voltage-gated calcium channels. So this is important, okay? Typically when we think of calcium, we think of our skeleton, we think of our teeth, and so on. But calcium plays a very critical role in a number of areas. Muscle contraction, which we'll see a little bit later, but here especially because you are not going to be able to conduct synaptic transmission without calcium. So calcium is going to move into the axon terminal, and what this is going to do, this actually causes the vesicles of neurotransmitter within the axon. Okay, so in here, okay, I've got these neurotransmitter, and what calcium is going to do, it's going to cause those vesicles to migrate, and then they're going to release the neurotransmitter through exocytosis, and they're going to move across the synaptic cleft via diffusion, going from an area of high concentration to low concentration. And then what's going to happen here is we're going to see, depending upon what that neurotransmitter is, it's going to change the properties. Here what's happening is I've got a voltage-gated sodium channel, and I'm going to have sodium come in, and I'm actually changing the electrical properties of that postsynaptic cell. Okay, so you want to be comfortable with these steps, the role of calcium, why calcium is important, the ultimate role that, once again, action potentials are necessary for the release of neurotransmitter. So this is a really important diagram, and you want to make sure that you're comfortable with it. Now, depending upon the postsynaptic cell and the neurotransmitter, that's going to have an impact on the action. Okay, there's lots of different types of neurotransmitters out there. We'll talk about some of them as we move through. Receptors, that's what the neurotransmitter will bind to. And what I want you to keep in mind here is it's the receptor that actually determines the change in cell activity once it's been activated. Also, we're going to be looking at different types of ion channels. All right. Now, those neurotransmitters that we call excitatory, what they do is they depolarize the postsynaptic cell. So what they're going to do is change the electrical properties of that postsynaptic cell to either exceed or approach threshold. An inhibitory neurotransmitter is going to be the exact opposite. And what that is going to do, it's going to hyperpolarize the postsynaptic cell. And that's going to make it more negative. Okay, so those are some really important terminology that you have to keep in mind related to how this all functions. Here is a list of a number of neurotransmitters. You may be familiar with some of them. Acetylcholine, that's going to be really important for muscle contraction. It's going to be really important in the autonomic nervous system. If we look at norepinephrine, okay, so that's very much tied into the sympathetic division of the autonomic nervous system. You may have heard of some of these, such as serotonin and dopamine, and so on. Another example would be endorphins. So all of these are examples of neurotransmitters, and they have some impact on the postsynaptic cell. Now, here we're looking at the integration and neural processing. And so generally, in the postsynaptic cell, there's a couple of things that are going on here. Okay, so how many neurons are forming synapses with it? And are those neurons forming synapses excitatory or inhibitory? Now, I want you to think about this pattern here, okay? Convergence versus divergence. So notice what's happening here. Think of this as input, right? I've got all of this input that's feeding into this one area. It's all converging, okay? Then we can kind of think of this as output. So now I'm diverging. So I have multiple effectors, and this is basically a very simple way in which how neural signals are processed.